Welcome to our video lecture on topic B32, transport in animals. This is our additional higher level content, and so as our friends, you will not be assessed against this. Our guiding questions continue to be, what adaptations facilitate transport of fluids in animals? We're going to skip the plants, which means we're also going to skip differences and similarities between transport in animals and plants. Our objectives for today, we're going to describe the function of some tissue fluids. We're going to outline structure and function of the lymphatic system. We're going to compare and contrast single circulatory systems and double circulatory systems. We're going to identify structures and functions of the human heart. We're going to describe adaptations of that human heart that allow for the efficient flow of blood. We're going to outline stages of the cardiac cycle and then read an ECG, also known as an EKG trace. In our previous video on animal transport, we talked quite a bit about capillaries. Here we're going to add some more details about tissue fluid. Tissue fluid can also be called interstitial or extracellular fluid. Extra because it's outside of the cells. Remember that our blood moves away from the heart in the arteries. Arteries branch into arterioles, which branch further into capillaries. These capillaries make up capillary beds that just give lots of surface area for these capillaries to be near all the cells of almost all of our tissues. We have a higher pressure in the arterial side of the capillary bed. That higher pressure allows for the movement of this fluid outside of the capillaries and into the tissues that are surrounding those capillaries. As that fluid, that tissue fluid, moves out of the capillaries and into the tissue, dissolved in that fluid, we've got lots of nutrients like glucose and sodium ions and amino acids. We also have some oxygen dissolved in that fluid, and this is how we're going to carry out that material exchange. We have our concentration gradients, so there's more glucose, more oxygen, more amino acids in the plasma of the blood, and that high concentration gradient is going to allow for the diffusion of the glucose, of the oxygen, of the amino acids out of the capillaries and into the tissues that are surrounding these capillaries. Now, oxygen is small and nonpolar, so it's going to move by simple diffusion. Glucose might need some help, which means that we probably have some facilitated diffusion going on here. Once the glucose, the oxygen, the ions of the salts are in the tissue fluid, those cells of the tissues might need to do a little bit of work to get the nutrients in, especially if we're talking about things like sodium ions or potassium ions, because these guys are so very polar, they're not going to move very easily at all through the plasma membrane of those tissue cells, and so there might be some kind of active transport, which means we've got some ATP going on. What's pretty cool is as we get toward the other end of the capillary bed, we have a lower pressure, and so a lot of that tissue fluid is actually going to move back into the capillaries, become plasma instead of tissue fluid once it's in the capillaries, and it's going to carry with it, dissolved in that tissue fluid now, are going to be some waste products and some carbon dioxide. So again, we can get all the good stuff out of the blood through this material exchange, the movement of the tissue fluid out of the capillaries into the space around the cells of the tissues. And then we can move the waste products, the carbon dioxide, back into the plasma of the blood so that it can get carried away. So lots of tissue fluid is going to move back into the capillaries and then get carried away by venules, those tiny little veins, and then the veins will carry it back to the heart. Sometimes there's a little bit of extra fluid that is not picked up by the capillaries at the venous end of the capillary bed, and we have this amazing lymphatic system right there to pick up that extra fluid and whatever other waste products need to be taken away from those tissues. Here we can see these lymphatic capillaries, which are very close to the capillaries of the circulatory system, and the lymphatic capillaries again are going to pick up that excess fluid. They're also going to pick up whatever other waste products are floating around that need to get carried away from the tissues. These lymphatic capillaries are going to merge into some vessels. These lymph veins are pretty cool. They have valves just like our circulatory veins have. and they're going to carry this fluid through the lymphatic system through some lymph nodes, which are pretty cool. So they're these just clumps, these masses of lymphatic tissue that have lots and lots of macrophages and lymphocytes, other blood cells of the immune system. And these lymph nodes are going to filter out bacteria and viruses, and sometimes they even catch some cancer cells. And so these guys are considered part of the immune system, not part of the circulatory system. The lymph, after it's been filtered out by these lymph nodes, is then going to dump this fluid into the circulatory system at the subclavian veins. The clavicle is our collarbone, that bone that sits here on the fronts of our shoulders, under our neck. There are veins under the clavicles, and so the lymph just gets dumped into that subclavian vein right before the blood makes it back into the heart. We humans and other mammals have a double circulatory system. We literally have two separate circuits. We have a pulmonary circuit. Pulmonary is for lungs, and then our systemic circuit is for the rest of the body. Here we have the right side of the heart. You're probably thinking, no, that's the left side. But if we're looking at the front of the heart, this is actually the right. So imagine that you turn yourself around with your back to the screen. This is the right side from the front. Into the right side of the heart, we have an atrium and then a ventricle. We'll talk about these guys in a couple more slides. So blood is going to move into the right side of the heart through that atrium, through that ventricle. The ventricle is going to pump the blood just to the lungs. This is moving at slightly lower pressure than what we'll have from the left side of the heart. We're carrying lots of carbon dioxide rich blood into the lungs. We're going to flip out that CO2. We're going to grab onto some oxygen, and then we're going to move back into the heart. So we're going to go to the left side of the heart with that oxygen rich blood coming from the lungs. We're going to go into the left atrium, the left ventricle. That left ventricle is going to give a big pump. We're going to move out through the aorta of the body, and then this oxygen-rich blood is going to be carried through the whole entire body. Here we have our beautiful capillary beds where we give that oxygen to the tissues, pick up some carbon dioxide that will then carry back to the right side of the heart, to the lungs, to the left side of the heart, to the body, to the right side of the heart, to the lungs. The brilliance of this double circulatory system is that we can maintain this super rich oxygen blood going to the body. So we don't have like, meh, it's kind of oxygen, but it's still got some CO2 as it's going to the cells of the body. It just makes for super efficient oxygenation of the blood. This is not true of things like fishes. So fishes do not have a double circulatory system. They have a single circulatory system. They have just two chambers in their heart. There's no left side, right side. There's just atrium and ventricle. The carbon dioxide rich blood coming from the body is going to flow into the atrium, into the ventricle. The ventricle is going to push it out through this artery because arteries always go away from the heart. But this artery is still high in CO2 and low in oxygen. This blood is going to get sent first through the capillaries of the gills. And in the gills, this is where the fish is going to exchange with the water that it lives in, that CO2, and it's going to pick up some oxygen that's dissolved in the water. That oxygen-rich blood will then flow around the body through the capillaries. Exchange will happen again. O2 out to the cells, CO2 back into the capillaries, and those capillaries will merge into those veins. Veins will carry the blood to the heart, and the heart will pump it again to the gills, where that gas exchange will occur. It's pretty amazing, these different adaptations that we have for different environments that we live in. So we mentioned those atria, those ventricles, the blood pumping into the heart, out of the heart. Here we're going to add lots more detail about that. So here we have a diagram of a human heart. This is the vena cava. The vena cava is the main vein of the whole entire body. We actually have what we call a superior vena cava, and here we have the inferior vena cava. It's not lesser, it's just lower. So this is the vena cava coming from the bottom, the lower half of the body. This is coming from the top half of the body. And they merge here at the right atrium. So the right atrium, this chamber of the heart, this room, this space inside the heart where blood will flow. So blood's going to flow vena cava into the right atrium. We're going to go past this super cool valve. This valve is called the right atrioventricular valve because it's between the atrium and the ventricle on the right side of the body. We're going to move from that right atrium through the right atrioventricular valve into the right ventricle. So here we are in the right ventricle. This is a slightly larger chamber of the heart. This guy's going to give a good pump. That blood is going to move this way past another valve. This valve is one of our semilunar valves. It's semilunar on the right side, so it's our right semilunar valve. We can also call it the pulmonary valve because it is going to control the flow of blood through the pulmonary arteries. These are arteries because they're going away from the blood. Pulmonary because we're going to the lungs. So the pulmonary arteries are going to carry blood away from the heart to the lungs so that we can trade out that CO2 and grab onto some oxygen. That oxygen-rich blood, I'm going to switch colors, is going to flow back into the heart through these guys. These are the pulmonary, because we're coming from the lungs, but because we're going to the heart, they're veins, pulmonary veins. Pulmonary veins are going to carry the blood from the lungs back into the heart, but now we're on the left side of the heart. That first chamber here is the left atrium. We're going to go past another valve. This is, you guessed it, the left atrioventricular valve, because again, it's between the atrium and the ventricle on the left side of the heart. From the left atrium, past that left atrioventricular valve, into the left ventricle. This is the strongest, thickest, most amazing chamber of the heart. This left ventricle is going to give a giant high pressure push. We're going to go through, there's this other valve that's kind of hiding back here. So there's another one of our semilunar valves. This one on the left side of the heart, so it's the left semilunar valve. We can also call this the aortic valve because we're about to push that blood into the aorta. The aorta is the main artery of the body. It's going to branch lots of branches and then carry that blood into arteries to all parts of the body except for the lungs. This whole cardiac cycle, this movement of the blood through the heart and out of the heart and back into the heart, is controlled by lots of pumps of the heart. So the heart is a pump. It's going to move to put force on the blood to push it around the body. So we have a couple different kinds of pressures that we talk about. We have systole and we have diastole. Systole is when we've got the contraction of the chambers. So here is the atrial systole because the atria are contracting. And then here we have the ventricular systole because now the ventricles are contracting. When we have this contraction, when the atria are contracting to push the blood into the ventricles, we have opening of the valves. We also have an increase in pressure because of that higher, that contraction, that bigger push. Here the ventricles are bigger and stronger, and so the pressure coming from the ventricle contraction is going to be a little bit larger. What's pretty cool, I'm going to erase some of my scribbles here. What's pretty cool about these pressure changes is that it causes the valves of the heart to open and close. So when the atria are contracting, when we've got this big push on the blood in the atria, it actually opens up the valves, those atrioventricular valves, so that blood can flow into the ventricles. At the same time, the valves, the semilunar valves, are going to stay closed, so the blood is kind of captured in the ventricles. When the ventricles start to contract, that ventricular systole, the blood pushing against these valves actually causes them to close, and then the semilunar valves open instead. So we're going to close off those atrioventricular valves. We're going to push open those semilunar valves so that the blood can flow through the pulmonary arteries and through the aorta. Kind of cool. Our two atria contract together, and those two ventricles will contract together. This contraction is myogenic. Myo means heart muscle, and then genic is like the beginning, the genesis of something, the generation. What's pretty cool, the heart beats itself. It does not need a stimulus from the nervous system to make it beat. The heart beats itself. There's this super cool little guy called the SA node here in the right atrium. This SA node, sinoatrial node, is a cluster of cells that stimulates the contractions within the atria. So the two atria will contract together because the SA node says, let's go and contract. The SA node also sends a signal to this guy, the AV node. The AV node is here between the atrium and the ventricle on the right side of the heart. This guy has lots of super cool branches of these cells so that we can send that signal out to the ventricles to get them to contract as quickly as possible. Contraction, again, is that systole. And then when the chambers relax, that's diastole. So we have systole, diastole for the atria. We have systole, diastole for the ventricles. That was a lot. Let's go ahead and review super quick. We've got blood moving into the heart, through the vena cava, into the right atrium, past a valve, into the right ventricle, past a valve, out to the lungs through the pulmonary artery. We're going to come back into the heart through the pulmonary veins, into the left atrium, past a valve, into the left ventricle, past another valve, and then out to the body through the aorta. We have a separation of the chambers, atria to ventricle, with these super cool atrioventricular valves. We also have a separation of left and right with this wall called the septum. All of these different chambers are going to have different pressures as different things are contracting and relaxing. Remember that contraction is systole, relaxation of the muscle is diastole. So here we're just looking at ventricular systole, ventricular diastole. That's going to relate to lots of pressure changes that are going to open and close those valves. Here we have our left ventricle's pressure. Notice, oh my goodness, it goes up so high and down so low. Crazy big force of that push on the blood. And then after the ventricle is empty, that pressure definitely releases a lot because there's just no blood to squeeze on anymore. And then here we've got that relaxation. And so that pressure is going to stay low as the ventricle fills back up. Notice that our atria, the left atrial pressure, stays pretty low. Atria just do not push on the blood with as much force as do the ventricles, which makes sense because they literally just have to get the blood from here to here. Not a lot of work being done there. Notice though that the pressure of the aorta stays crazy high throughout the cycle. This is because the aorta and other arteries have a very thick wall of smooth muscle that contracts down on the vessels to maintain high pressure of the artery blood. We also have lots of volume changes. So this part of the graph is just looking at volume, just of the left ventricle. It fills up, it empties, it starts to fill back up. EKG or ECG, we're going to talk about in a couple slides. This is just related to those nerve impulses that stimulate the contractions of the different chambers of the heart, the atria, the ventricles. And here we have the sounds, that lub-dub of the heart. The sounds that we hear, that heartbeat sound that we hear, is actually the closing of the valves. So as the atrioventricular valves close and those semilunar valves close, this is what we hear. If you want to watch a lovely animation of heart blood movement, contractions, and the movement of the blood through the heart, the opening and closing of the valves, this one is pretty great. We're going to watch it in class, but if you want to watch it now, it's kind of fun. I mentioned a few slides ago that heartbeat is myogenic. The heart beats itself with that sinoatrial node, which causes the two atria to contract. It also sends a signal to the atrioventricular node. We can also call these the SA and the AV nodes. The atrioventricular node, the AV node, causes the ventricles to contract together. The brain, the medulla of the brain, which is here in the brainstem, can change the rate of heartbeat. So it doesn't cause the heart to beat, but it can stimulate the heart to beat faster or slower. The sympathetic nerve, also called the accelerator nerve, causes heart rate to increase. The vagus nerve, which is part of the parasympathetic nervous system, causes heart rate to slow back down. So if I need to do lots of running, or if I have too much CO2 in my blood, then signals are sent to the heart to speed up, not to beat, but to speed up. When we're taking a nap, or we're sitting in class, or our video lecture's getting kind of boring, then the vagus nerve tells things to slow back down and go take a nap. We also have some hormones that can have an effect on heart rate. So our adrenal glands sit on top of our kidneys, and they make adrenaline, which we can also call epinephrine, that causes an increase in the heart rate. The opposites are noradrenaline, oh my gosh, I cannot spell. Hold on. Noradrenaline and norepinephrine. And these hormones are going to cause that heart rate to slow back down. So again, the heart beats itself. It is myogenic, but the brain and hormone system can have an effect on the speed at which we beat our hearts. We can trace that myogenic electric activity that causes the heart to contract using ECGs or EKGs. They're the same things, and this stands for electrocardiogram. Electro, because we're looking at the electric signals of these impulses that lead to the stimulation of the contraction of the heart. Cardio, because it's a heart. Gram, because it's a graph. So the first little part of our EKG trace is the P wave, and this is where the atria is depolarizing. We're going to talk about depolarization of nerve cells in a couple more lectures, but this is just that signaling of the nerve impulses through the cells to get the atria to contract. So that P wave sets off that contraction of the atria. We then have what's called the QRS complex. The QRS complex is going to cause the ventricles to depolarize. That sets off the systole contraction of the ventricles. The T wave is this tiny little wave that follows, where we have the ventricle kind of going back to normal so that it can contract all over again. Let's practice that a little bit more. Here we have our normal echocardiogram trace. I've got a P wave, that's the atria getting ready to contract. We've got our QRS complex that stimulates the contraction of the ventricle. And then we've got our T wave, that's the ventricles going back to normal so they can contract all over again. We've got our P wave, atria contracts. We've got QRS complex, ventricles contract. T wave, ventricles get ready to contract again. P wave, atria contracts. QRS, I got ahead of myself. QRS complex, the ventricles contract. T wave, ventricles are back to normal so that we can do it all over again. This is known as atrial fibrillation, where we are going a little bit wackadoodle. Notice that there is not a lovely regular trace. Notice that we don't have that lovely P wave anywhere to kind of control things to get it to go. So this is bad. This is irregular heartbeat. How do we fix this? We shock the heart back into a normal rhythm. Sometimes we call it the normal sinus rhythm. But this is bad, and this is why we have our defibrillator machines around the school so that we can help people who are struggling to maintain a regular heartbeat. And on that note, my friends, we have arrived at the end of part 2 of animal transport. We talked about tissue fluid, which we can also call interstitial fluid, that juicy stuff that has all those good materials for material exchange between the capillaries, the circulatory system, and then the tissues. We talked about the lymphatic system. It's going to pick up some of the extra fluid from around the tissues, carry it through lymph nodes that filter out bacteria and viruses and sometimes even some cancer cells before that fluid gets dumped back into the circulatory system. one. We talked about single circulatory systems that the fishies have with their gills, and then our double circulatory systems where we have pulmonary to the lungs and systemic to the rest of the body. We talked about those amazing structures and functions of the human heart. We have a left side, a right side, we have atria, we have ventricles, we have lots of valves. We talked about adaptations, how we have some different pressure changes. We have valves to prevent backflow. We've got the left side and the right side so that we can have not just efficient blood flow, but efficient flow of oxygenated blood from the lungs. We talked about those stages of the cardiac cycle, the systole, the diastole, and then we looked at our lovely EKG or ECG trace. Great work today.